Hi, rabbits. Research Turtle here. Today, we are taking a molecular deep dive into a fascinating paradox in modern nephrology. We are looking at how SGLT2 inhibitors, drugs designed to dump glucose in the proximal tubule, somehow manage to save magnesium in the distal tubule. If you look at the kidney map, you'll see the disconnect. SGLT2 blockade happens early in the nephron, yet the magnesium retention happens downstream at the TRPM6 site. This isn't just a side effect, it is a reversal of the wasting we typically see in diabetes. Before we get to the how, let's remember why. Magnesium isn't just an electrolyte, it is the engine oil of life. It stabilizes ATP, drives DNA synthesis, and powers over 600 enzymatic reactions. In clinical practice, hypomagnesemia is dangerous, driving insulin resistance and cardiovascular instability. The fact that SGLT2 inhibitors consistently elevate serum magnesium is a massive collateral benefit that we need to understand. Meet the gatekeepers. TRPM6 and 7 Chanzymes. These aren't standard ion channels. They're unique Chanzymes, a fusion of an ion channel and an enzyme kinase on a single protein chain. Here's the critical rule. TRPM6 is the magnesium specialist found in the kidney and colon, but it's socially awkward. On its own, it gets trapped in the cell's endoplasmic reticulum. It must pair up with its cousin, TRPM7, the housekeeper, to form a heteromer. Only together can they travel to the cell surface and do their job. So how do SGLT2 inhibitors help? We've identified 3 distinct pathways. First is the insulin pathway. In uncontrolled diabetes, insulin resistance breaks the signaling chain. The RAC1 protein goes dormant, and TRPM6 vesicles get locked inside the cell. By resolving glucotoxicity, SGLT2 inhibitors restore insulin sensitivity. This wakes up RAC1, which grabs those TRPM6 vesicles and rushes them to the surface. It's a restoration of cellular traffic. The second mechanism involves glucagon. SGLT2 inhibitors trick the body into a fasting-like state, slightly raising glucagon levels. This glucagon binds to receptors in the distal tubule, triggering a CAMP and pKa cascade. This kinase directly phosphorylates TRPM6 at a specific spot, residue S1252. This chemical modification locks the channel open, forcing magnesium uptake even if insulin signaling is still weak. The third mechanism is fascinating and happens in the gut. Some inhibitors, like canagliflozin, transiently block SGLT1 in the intestine. This leaves a bit of glucose behind, which bacteria ferment into short-chain fatty acids. This drops the local pH. Now, look at the pore of the channel in the diagram. It has specific glutamate residues, E1029 and E1024. These are proton sensors. The acidic environment protonates these residues, essentially jamming the door open for maximum magnesium absorption. There's also a structural bonus. In models of kidney injury like cis-portin toxicity, the distal tubule usually atrophies, it shrinks. SGLT2 inhibitors prevent this. They preserve the physical architecture of the tubule, keeping the TRPM6 machinery intact. Now let's look at the broader regulatory landscape. SGLT2 inhibitors aren't the only players. We have natural allies. EGF is the most potent activator. Blocking it, like in cancer therapy, causes severe magnesium loss. And estrogen explains why premenopausal women have higher magnesium levels than men. It boosts TRPM6 transcription and removes inhibitory proteins. But we also have enemies. Look at proton pump inhibitors, PPIs. By blocking stomach acid, they alkalize the gut. Remember our proton sensors? In an alkaline environment, those residues lose their protons and the channel closes tight. This is the PPI paradox. Then there are transplant drugs like cyclosporin. They work by silencing the immune system, but they also silence the gene transcription factor NFAT. Without NFAT, the kidney simply stops making TRPM6. To wrap up, look at the summary matrix. SGLT2 inhibitors stand out because they hit multiple targets, they fix insulin sensitivity, they utilize the glucagon pathway, and they protect the kidney structure. This transforms our understanding of these drugs. They're not just lowering sugar, they're metabolic regulators restoring electrolyte balance through elegant molecular crosstalk. That's it for today. Turtle out.